Hello everyone. So in this third video, we are going to explain about the basic amino acids of proteins, their structure, and we will also discuss about the components of nucleic acids. So some of the amino acids of proteins are alanine, serine, aspartate, tyrosine, histidine, and cysteine. So these are some of the important amino acids which whose structure we have to keep in our mind, and we need to practice these structures, but we need to remember a basic framework of these amino acids which is basically we can start it from here alanine. So as we can see here, it has been mentioned that the basic framework which is C, one side is H attached, one side is NH3 plus, and COO minus. So CH NH3 plus COO minus will be remain the same in all the amino acids, basic amino acids. But what will change will be the functional group. So here in alanine it is CH3. Methyl group is attached, CH3. Then what in serine we have to remember that the same framework C COO minus is above, NH3 plus is here, H is there, but CH2OH is attached. In alanine CH3 was attached, in serine CH2OH is attached. Then we can remember of aspartate. So aspartate, again this basic framework is the same. The functional group is CH2COO minus. So in alanine it is CH3, in serine it is CH2OH, and in aspartate CH2COO minus is there. Now in tyrosine, again in tyrosine, and one more common thing we can see that in alanine when we divide this CH3 into further, when we straighten it, we write down, we break it down, so it gets broken into CH2 bond H. Again here in serine CH2 bond OH. Again here in aspartate CH2 bond COO minus. And in tyrosine again the functional group is CH2 bond. This is the benzene group, and OH is there. CH2 this benzene group and this OH group. In histidine, again this group, this is the functional group, and in cysteine, CH2SH is there. So we need to remember these amino acids of proteins uh, by remembering the basic framework and then attaching the functional groups. We can practice it one or two times, and we can keep it forever in our minds. And then we will see the component of nucleic acids, which are uracil, thymine, cytosine, adenine, and guanine. So these are the nitrogenous bases of, for ribosomes it is uracil, thymine, and cytosine, and for uh, this for DNA it is adenine and guanine, AG, CUT. So CUT for cut and AG. So we can remember these, and we can remember the structure. As we can see in uracil, thymine, and cytosine, what is changing, what is the change in the structure. So we can see that uracil and thymine, here this here it is in thymine CH3 is connected with this carbon group, but in uracil only H is connected, so there is difference of hydrogen bonds here. So in rest of the structure is same in uracil and thymine. So thymine extra hydrogen is attached, we have to remember this, and rest of the structure is same as uracil. Similarly, in this cytosine, one of the H has been removed here from nitrogen, and rest of the structure is, we can see, is similar to that of uracil. So uracil is, we can understand that uracil, we can take uracil as one of the basic structures where the NH, when it differs from the cytosine in H, from lacking in cytosine, and in thymine it is once extra H is there, extra two H are there. So uracil, thymine, and cytosine are can be remembered by this way, and adenine and guanine we can remember the structure. As we can see, what is the difference is that at the place of here O, here NH2 has been attached in adenine, and in guanine the OCO group is there, and rest of this in rest of the structure in guanine extra NH2 group is there, but in adenine there is no NH2 group. It is CH only. So we need to remember these points, like in guanine NH2 group is present, and if we are given the structures we can identify, because generally in the questions the structures are given and we need to remember. In the multiple choice question we need to remember that which of the nitrogenous base is it. So using this formula and practicing 1 to 2 times we can remember these structures definitely. So now these are 5 carbon sugars. One is alpha D ribose and one is 2 deoxy alpha D ribose. So what is the basic difference between these two structures here? In 2 deoxy alpha D ribose, downsides one O is lacking, and in alpha D ribose OH is there, here H is there. So this is the basic difference. So this also we can remember. So alpha D ribose this is, and this is 2 deoxy alpha D ribose. So we can see that in 2 deoxy alpha D ribose, at the second position O is not there. So it is 2 deoxy, but means at the second position oxygen is lacking. So in this video we will discuss up to this. We will go to cells build supramolecular structures in our next video. Thank you very much for watching the video. Keep watching. Thank you.